A very good morning to one and all. I extend a warm welcome to our distinguished guest for the day, Dr. Kumar Venkitanaran, Professor and Associate Dean for Research, University of Connecticut, Seoul, USA, our beloved Dean, Dr. V. Sayan, Faculty of River, PG scholars, undergraduate students, and non-teaching staff. Today we have gathered here to have a talk session on antimicrobial resistance in poultry. Before we begin, I request our beloved Dean, Dr. V. Sayan, to welcome and honor our chief guest, Dr. Kumar Venkitanaran sir, with a bouquet and memento.
Sir?
I'm profusely elated to take an opportunity to introduce our chief guest of the day, Dr. Kumar Venkitanaran, Professor and Associate Dean of Research, College of Agriculture, Health and Natural Resources, University of Connecticut, Seoul, USA. Dr. Kumar Venkitanaran sir received his Bachelor of Veterinary Science and Master of Veterinary Science degree from Kerala Agricultural University and Tamil Nadu Agricultural University, India, respectively, after which he practiced as a large animal veterinarian for a year. He obtained his MS in Food Science and Technology in 1992 from the University of Nebraska, Lincoln, and PhD in Animal Science in 1996 from the University of Connecticut. From 1996 to 1999, he worked as a postdoctoral research associate at the Center for Food Safety, University of Georgia. In 1999, he joined the Department of Animal Science, University of Connecticut, as the assistant professor. He currently serves as the Associate Dean for Research and Graduate Studies at the College of Agriculture, Health and Natural Resources, University of Connecticut. His research investigates the potential of plant-derived molecules, essential minerals and probiotics for attenuating virulence in human and animal pathogens. He has published 134 peer-reviewed journal manuscripts, 31 published book chapters, 129 abstracts, and characterized 5 new bacterial genes. Having successful in garnering over $18 million as funding for his research, Dr. Venkitanaran has mentored over 85 candidates in his laboratory, including 18 PhD, 10 MS, 5 postdocs, and over 55 undergraduate students in research. He currently serves as an editor in PLOS ONE and as an editorial board member in 4 other journals. Dr. Venkitanaran is the recipient of several accolades, including elected as the Fellow of the Institute of Food Technologists, Evelyn Degusta Award by the U.S. Poultry Science Association, Food Microbiology Division Award by the Institute of Food Technologists, and Research Excellence and Creativity Award by the University of Connecticut. Without further ado, let's begin our today's talk on antimicrobial resistance in poultry. I'm honored to invite Dr. Kumar Venkitanaran sir to come on stage and share his valuable insights.
Okay, let's get started. Honorable Dean Dr. Sayan, faculty members, and my dear students. As I always say, it's really exciting to be visiting River, because as you know, I visited an institution just 3 months ago. And I used to tell you, Dean, it's like coming back to my own home or alma mater, because I have developed such a relationship with this institution, including past students, friends who serve as faculty members, etc. So, without taking time, I would like to get started with my presentation. And I really appreciate all the interest from the students. That's a driving force for me to come back again and again. So, the topic I'm going to talk today is, as you can see, how to control antibiotic resistance in poultry operations. Your institution, I know, have been working on the same topic in the poultry industry. And this project is part of a huge USDA project I have, and one of the objectives. And why I selected this topic is because we are I'm talking with your dean for a potential collaboration on a project on this topic. and I wanted to give the perspectives which I am following in targeting antimicrobial resistance. So whenever I talk about antibiotic resistance, I always recognize or give credit to Dr. Alexander Fleming. Historically, we all know that the discovery of penicillin by Dr. Alexander Fleming marked an important era in public health, which was followed by identification and manufacture of a variety of antibiotics, which includes natural as well as synthetic antibiotics. So this was followed by enhanced infection control and extended life expectancy globally. Then sometime during the 1940s, it was recognized that a number of antibiotics, when supplemented at subtherapeutic concentrations to food-producing animals, they increased the feed efficiency and accelerated growth. This sparked widespread use of a wide array of antibiotics in animal agriculture or food animal production, and I would say that subsequently the extended use of antibiotics for a long period of time, which includes use, abuse, and misuse of antibiotics, not only in animal agriculture as therapeutic agents in veterinary medicine and also in human medicine, contributed to the global issue of antibiotic resistance. And then once we recognize this as an emerging public health hazard, many countries have come up with strategies. Some of them have even banned, especially Scandinavian countries, have banned some of the antibiotics in food animal production. Many countries are phasing out antibiotics from animal agriculture, including US. The FDA has regulations to phase out antibiotics. And the quest for alternative to antibiotics, especially as growth promoters, is on the way. So with that introduction, let me begin my talk. And one more thing I wanted to tell you, which I read in a journal, when Alexander Fleming discovered penicillin, a few months after that, his lab notebook indicates that he already found bacteria resistant to penicillin. So the rapidity at which microorganisms develop resistance is phenomenal. So antibiotic resistance, I don't have to give any more introduction. It is indeed a global public health hazard now. A report published by CDC from the US in 2019, that was the extensive report, which shows that in the US we get around 2.8 million illnesses due to antibiotic resistant infections, which has a total of 35,000 mortality. The same report projected that by 2050, antibiotic resistant infections would be the #1 cause of mortality in human beings. And also the same report estimated that the economic impact due to antibiotic resistant infections and mortalities include hospitalization, medical costs, loss of productivity, loss of life, everything equated to almost $100 billion. So what are I told you, antibiotics are used in human medicine, antibiotics are used in veterinary medicine, and from a therapeutic point of angle, then a wide variety of antibiotics are used as growth promoters, which has a huge economic angle for the producers. In addition to that, some of the factors we need to consider when we are using antibiotics is a lion's share of antibiotics administered to humans and animals, especially animals, they are excreted in the urine and feces. And so they are recycled back to the environment, whether it's soil, water bodies, everywhere, and bacteria get exposed to those antibiotics, so more generation of antibiotic resistant bacteria happens. Similarly, in the US, we have when we have an antibiotic, and people, when we have an excess antibiotic after our course, there will be a container and an envelope. You put those antibiotics in the bottle, put it in the envelope, and you can return it to the pharmacy. Just don't flush it in your toilet. I don't know how many countries would practice that, but that is another concern, because improper disposal through the toilet or any sewage systems, finally the antibiotics end up in surface waters. Then it holds the environmental, the whole environmental transmission. Animals bring it, that could be used for agriculture, so it spreads the antibiotic resistant bacteria and the antibiotic resistant genes that exist there. And then one other topic which is often neglected is, many of us we may not recognize, is that a large amount of antibiotics are used in agriculture, crop production. They use it for spraying against diseases. And in the US alone, we used, this was a 2019 report, we used around 70,000 kilograms annually. And most countries, when I looked at it, they don't have any regulations or guidelines on antibiotic usage in crop production, although they use tons of antibiotics in crop production. Coming to the antibiotic resistance in India, I was talking to Dean Sejin. Last week I was at Kolkata, National Institute of Cholera and Infectious Diseases, and I was talking with the deputy director there who works with ICMR very closely. So based on my interactions, I see that there was a report in 2029 published by ICMR, and the control measures to target and the AMR in India are in the beginning stage. And what we have seen is, when we address antibiotic usage in humans and animals, there's a term called DDD. It is defined daily dose. If you compare now, compared to two major other countries, China and India, as you can see, India has the highest daily dosage, 6.3 billion DDDs, compared to China, 3.8, or US, 2.8 billion. This is, can be contributed heavily to the free availability of antibiotics in India. That report says that it's due to the free availability, partly it is due to the free availability of antibiotics in this country. And another aspect this report said was, again, the environmental presence of antibiotic resistant bacteria and genes, especially in surface water bodies, actually. The report finally said that there's a 30% increase in antibiotic resistant births over the last so many years in India, and most of the therapeutic antibiotics are not able to inactivate multi-drug resistant pathogens. And as I said, efforts to control AMR are in very preliminary stages. I believe the government of India is trying to address it, putting together guidelines and all. So when it comes to antibiotic resistance, a lot of efforts are being undertaken. Mostly, the first one that comes to every country is reduce the use of antibiotics. Of course, antibiotic usage has to be, especially without a purpose, just the injudicious use of antibiotics has to be controlled. It has to be tightly regulated. So that's, of course, that is needed. And then, in terms of controlling antibiotic resistance, there are many people do surveys, prevalence studies to look at the prevalence of antibiotic resistant bacteria in the food production chain. Of course, they are also important because you need to understand the magnitude of the problem. Only then you can devise control strategies. But one thing, my research is on a totally different perspective, which I'm going to share with you. Bacteria are very smart microorganisms, and they have a remarkable ability to share their genetic information when their survival is challenged or threatened. So they share genetic elements like virulence genes, survival traits, and antimicrobial resistant genes among them by a process called horizontal gene transfer or horizontal transfer of genetic elements. There are three different methods of SGT, which now I will say that SGT, which represents horizontal gene transfer. Transformation is one method by SGT happens. What do we know about transformation? It's simple uptake of antibiotics from the environment by the bacteria, from the environment where they are. It's basically uptake through the cell wall. The second one is transduction, which is basically virus play a role. Virus transfer genes from one bacteria to another bacterial cell. That's called the transduction. But the most common method of horizontal gene transfer, especially transfer of antimicrobial resistant genes, is conjugation. So here you can see conjugation is a process when two bacterial cells, a donor and the recipient, come together. Then they establish a conjugative bridge through a hair-like appendage seen on bacteria called bacterial pilus. So the pilus is very critical to form a conjugative bridge to establish a connection. So once the conjugative bridge is formed, the genetic material is exchanged, the recipient receives the genes from the donor. So this is the most common method of dissemination of antibiotic resistant genes in bacteria. You need a donor and a recipient cell for that. So in controlling antibiotic resistance, simple reduction in the use of antibiotics would not solve the problem. That is because antibiotic resistant bacteria, whichever environment they are, they multiply, evolve, and maintain the resistant traits, the genes, even there is no antibiotic. So even if you don't use antibiotics, there is no antibiotic, these resistant genes have been already created for the last so many decades of use. So the resistant bacteria are there and the genes are there. And they keep disseminating from one bacteria to another, even in the absence of antibiotics. So to give you an example, I believe in the 1980s, chloramphenicol resistant, chloramphenicol was banned from animal agriculture. But there are studies that even after that, the farm environment yields E. coli resistant to chloramphenicol. So that means once they gain the resistance, they keep transmitting or spreading the resistance to other bacteria as a chain reaction. So we cannot reverse bacterial resistance by simple reduction in antibiotic use. Don't get me wrong, I'm not projecting that or recommending that this is not going to be useful. Of course, we have to reduce the use of antibiotics. But that alone is not going to control the issue of AMR. So we need novel approaches to break that chain or inhibit the horizontal gene transfer of antibiotic resistance. So the project which I'm especially going to share with you is a few projects on this HGT. Today what I'm going to present to you is we use certain phytochemicals we have identified which disrupt the horizontal gene transfer so that the transfer of resistance is inhibited from bacteria to bacteria. So what are the hotspots for horizontal gene transfer in a poultry production environment? Of course, the birds itself, in the gastrointestinal tract of birds, in all animals it has been reported, including human, horizontal gene transfer can happen from one resistant bacteria to a commensal bacterium, and from that to another bacterium. So that can happen in the poultry gut. So that we should target that, that's a hotspot. And in fact, there are studies which have shown that animals which have never been given any antibiotics, their manure will still have antibiotic resistant bacteria. How? Because of the process of dissemination. They get the genes from, uptake the genes, get the genes from others, bacteria or their surrounding environment. Another hotspot is manure. It's the high incidence of HDT has been reported. And water bodies, surface waters, water levels in the poultry environment or farm environment at another source. So in terms of, we know that in poultry production, I was talking with the faculty just before this talk, we had a discussion on AMR and poultry. The major foodborne pathogens include Salmonella and Campylobacter, which contributes to together about 70% of the foodborne diseases transmitted through poultry products. So, between Campy and Salmonella is of course more antibiotic resistant and highly prevalent. And the test bacterium we use in my research is Salmonella heidelberg. Salmonella heidelberg is a serotype of Salmonella which is transmitted through poultry products in the US. It is characterized by high invasiveness in the host and also resistant to multiple antibiotics, which are seen here, especially beta-lactam group of antibiotics. We know beta-lactam group of antibiotics, penicillin, ampicillin, amoxicillin, which are the most commonly used antibiotics in human and veterinary medicine. They are resistant because they have a gene or rather a plasmid called blatam. The blatam plasmid confers resistance to beta-lactam group of antibiotics. So we targeted this bacterium because this could transfer its genes from its cell to other bacteria, especially commensal bacteria in the poultry production environment. So we wanted to see whether we could disrupt that dissemination. So phytochemicals, my lab has been working with phytochemicals since 2000. We all know that, plants produce a variety of phytochemicals, almost up to 7,000 phytochemicals have been identified. And these phytochemicals have been reported even from the times of Hippocrates. And then the Native Americans have used it for some purposes. And also Ayurveda, we shouldn't forget Ayurveda, which uses plant constituents, extracts for improving human life, promoting human health. And then it's been used in many cultures as food preservatives, flavor enhancers, etc. And why phytochemicals? As I told you, my lab has been working for almost 20 years on phytochemicals. We have screened more than 80 phytochemicals in the lab. What we have found is there are a few phytochemicals which have been very effective in controlling Salmonella and Campylobacter. I told you the two major foodborne pathogens in poultry when we use it as a feed supplement or additive. And we have found that up to certain concentrations, the phytochemicals have no adverse effect or toxicological or adverse effect on the production parameters like growth, egg production, etc. But you should remember that I was just sharing with the dean and others, the birds, we expect the birds to eat anything, but I told you no adverse effect until that concentration, because if it is a high concentration, the birds will starve for weeks and die rather than eating the diet containing a high concentration of the phytochemicals. So we have to be extremely careful. And most of the phytochemicals are classified as generally regarded as safe or food grade by the US FDA, and they are environmentally safe. So as I told you, we have been worked with many phytochemicals, and we use a rigorous procedure for finally when we select them to put them to use in vivo systems, in birds or animals. So we have libraries to screen the phytochemicals. So based on the structure, the library will give us a cue or an indication what would be the targeted site of the action of these phytochemicals. So based on that information, we select phytochemicals for different purposes. If it is to attack the colonization factors in bacteria. Based on the screening results, we use different types of phytochemicals. Or if it is to attenuate virulence, we use different set of phytochemicals based on the screening results. So once we select the phytochemicals based on the screening, we use broad dilution studies to check their efficacy. Then we resort to cell culture to validate our results. We also check for quorum sensing or virulence gene expression. Quorum sensing, I don't know how many of you know. Quorum sensing is basically a communication mechanism among bacterial cells through a production of a messenger molecule. So it's like a cell phone signal, you guys all talk to each other. Bacteria produce that messenger molecule, and that is sensed by all the bacterial cells in an environment. Then that is a signal they get. So what they do is once bacterial number in a host reach certain population, they send the messenger molecule, which is sensed by the quorum sensing mechanism. And then they all say, okay, we have reached a quorum, we have reached adequate number. Now let us enhance our toxins production, enhance our gene expression, so they attack the host. So we looked into the efficacy of phytochemicals to disrupt the quorum sensing. Then we use an invertebrate model. Then finally we go for a mammalian or a avian model. So these are the rigorous steps we follow. And the phytochemicals we used in this SGT experiments, we screened about 15 to 20, only 4 of them were effective. One is panthenamyl, here on which I will use the acronym TC, which is a major ingredient in cinnamon oil. Carbachol is ingredient in oregano oil. Caprylic acid is an 8-carbon fatty acid present in coconut oil. And also the milk of goats, we can relate the caprylic acid, caprines. And then beta-racosalic acid is a phytochemical seen in cranberry juice. So the phytochemicals in our lab, when we wanted to address the virulence, not to kill the bacteria, because when we use any antimicrobial that would challenge the survival of a microorganism, the bacteria especially, they would, after certain generations, they are smart enough to mount resistant mechanisms. That's how we get the antibiotic resistance. So if you challenge the existence or survival of a bacterium with a molecule, first time you will be able to kill, second time you will be killed. After a few generations, they will have genetic mechanisms that will bring resistance to the molecule. So we don't use high concentration which will kill or inhibit the bacteria. We use SIC, which means sub-inhibitory concentrations, which have no effect on the bacterial cell from killing them, but their concentration that modulates gene expression and virulence. So they basically reduce the virulence of the genetic mechanisms. So here, the genetic mechanisms that contribute to the horizontal transfer. So we use concentrations of phytochemicals, those phytochemicals which will disrupt the genetic mechanisms that will result in the HGP of antibiotic resistance. That's the whole purpose of this study, and I will show you what data we get. So we have to first determine the subunitary concentration of all these phytochemicals. We determine in LB, first we have to do a broad dilution, so we do a lab medium. Then, as I told you, the two major environmental sources, chicken manure and water, for the hotspots of the HGP. We determine the concentration, SIC of all those 4 molecules in these 3 media. Then, as I told you, for the HGP experiments, as I told you, the Salmonella Heidelberg, which is highly resistant to antibiotics, because it has a lactam plasmid, which contributes resistant to beta-lactam antibiotics. We use it as a donor. So because of the lactam plasmid, it is inherently resistant to ampicillin. And the donor, the recipient, we use the commensal E. coli, because our principal objective is to see if the phytochemicals will disrupt the horizontal gene transfer of resistance from the resistant salmonella, which is very common in poultry, to a commensal E. coli. And we induce the resistance to naldixic acid in E. coli, because we wanted to use as a marker to distinguish the recipient bacterium from the donor and other bacteria. So horizontal gene transfer experiments, now the principle is we do the experiment, we grow them together. What happens is a conjugate difference is formed, and then from the donor, the black-gum gene is exchanged, is transferred to the recipient. So the E. coli recipient is inherently induced for resistance to naldixic acid. When it gets the black-gum plasmid, it becomes resistant to ampicillin. So now it is called as a transconjugant. So transconjugant is one which receives the gene or a plasmid from the donor and becomes resistant to both ampicillin and inherently and also to naldixic acid. So how do we confirm the transconjugant once we receive the gene through HCT? You can see here donor which has a ampicillin black-gum plasmid, it is resistant to ampicillin, so it grows on plates containing ampicillin. It doesn't grow on naldixic acid because we did not induce resistance, but it grows on plates containing naldic acid, and it doesn't grow in plates containing naldic acid or ampicillin. But the recipient E. coli, we induce, it doesn't have any ampicillin gene. So it doesn't grow in ampicillin, presence of ampicillin, but we induce resistance to naldixic acid, so it grows resistant on plates containing naldixic acid, but it doesn't grow on plates containing both because it is not resistant to ampicillin. Whereas the transconjugant, the E. coli which became a transconjugant when it received the gene through HCT, it is already resistant to naldixic acid. Once it gets ampicillin, it is resistant to ampicillin. The black-gum plasmid is resistant to ampicillin also. So the transconjugant grow in ampicillin, in naldixic acid, because the recipient became a transconjugant and then both these. So this is how we confirm the transconjugant after the SGT, and we also do the PCR to identify the genes. So these are the first experiment is looking at the effect of phytochemicals on inhibiting HTT between Salmonella and E. coli in different media, LB, manure, water. So we use the published protocol where we inoculate equal population of the multi-drug resistant Salmonella Heidelberg, commensal E. coli. We add the subunity concentration of the phytochemicals separately, incubate them, plate them, and determine the transconjugant. Transconjugants are those, once they receive the HDT plasmid, they are resistant to both naldixic acid and ampicillin. And then we use a published, very commonly used formula where we determine the HDT frequency for the control group, the treatment group, then we find a ratio and multiply it by 100 to get the determine the relative HDT frequency. So these are the results. This shows the effect of phytochemicals in reducing HTT between Salmonella and E. coli in LB broth. The x-axis are the different treatments. Y-axis is the relative HTT frequency. The HTT frequency in the control sample, which is dark, is taken as the 100%, and that in the treated samples are expressed relative to that. As you can see, compared to the control, all the four phytochemicals significantly reduce the HTT frequency in the treatment. Similar to that, we determine the same HTT frequency in manure and water. We found the same results in poultry manure. All the four treatments substantially reduce the HTT compared to the control in poultry manure, and similar results in water. Then now we determined that it could disrupt the HTT transfer of antibiotic resistance from the multi-drug Salmonella to commensal E. coli in the environment, especially poultry manure and water. Now we wanted to see whether it would be effective in inhibiting the HTT in the animal gut. As I told you before, HTT has been shown to occur in animal gut, human gut, and they could, so if it happens in the human gut, it could transfer the resistance gene to millions of bacteria, including the normal flora, probiotic bacteria, because I was talking to your dean actually. I had a faculty from Tamil Nadu Veterinary University just visited my lab for 3 months. She did a project continuation of this. she looked into whether probiotics could receive the antibiotic resistant genes from the resistant bacteria and could transmit to other bacteria. What she found is probiotics, the resistant gene from Heidelberg is transferred to probiotic bacteria, then the probiotic bacteria to commensal bacteria, and this happens in milk, in meat, in refrigeration temperatures. So raising a big concern actually. We are very preliminary in that because she could spend only 3 months, so we will be doing follow-up studies on that. So we have different groups in chicken. We have a negative control where we did not feed anything, positive control where we fed the birds with the inoculated the birds with the Salmonella Heidelberg and the E. coli. Then the same we have the birds which were inoculated. We feed different concentrations of panthenol and carbachol. We selected these two because they were found to be more effective in the in vitro experiments, and also the birds have a better palatability for these two hormones. So the ex vivo study was done in published model. We obtained day-old chicks, fed them with the phytochemicals from day 0. Then on day 7 and day 16 after feeding, we sacrificed the birds, collected the cecum because cecum is the hotspot where bacteria salmonella colonize and also HGT occurs. So we collected the cecum, injected with 8 log equal portion of multi-drug salmonella and E. coli, incubated them, serially diluted, enumerated transconjugates and determined the relative STT frequency. So these are all the results on day 7. Again, relative STT frequency, different treatments. As you can see, compared to the control, multiple actually all of them reduce the STT frequency, but especially 1% TC, 0.124% CR, and a combination of CR and TC, the difference was significant statistically. Similarly, the results on day 16, we found similar results in parallel with the previous slide or day 7. And now, from ex vivo, we want to go to the in vivo study. The only difference here is we did not collect the cecum, but we fed the birds with the phytochemical and orally inoculated the donor and recipient, the Salmonella Heidelberg and E. coli. Then sacrifice the birds day 10, 12, and 14 and determine the relative frequency. So these are all the results you can see again on day 10, day 12, day 14. Compared to the control, there was a consistent trend in the reduction of HGT frequency, but not all the treatments were statistically significant. And another aspect is you can see the standard error. How this is because these are live birds, right? They have other antibiotic resistance. Although we screen before using, they pick up antibiotic resistant bacteria from the environment. So that's why we have a huge error bar here. So now switching gears, we found out that this, especially the couple of compounds consistently were very effective in preventing the HGT. So we were curious how it is bringing out the effect, the mechanism of action. As I told you in my initial slide, when the bacteria form a conjugative bridge, they need a hair-like appendage like villi, which forms the conjugation bridge. So it is known that villi are important for conjugation, and also mutations in the genes that transcribe the villi. Once mutation happens, conjugation doesn't happen. So we know that they play a significant role from published literature that villi plays a role in HTT or conjugation. So what we did was, these are the genes. There are several genes which are critical for villi formation and conjugation. These include, they are called the TRA genes or transfer genes. There are a number of transfer genes. Once their expression or transcription is decreased, conjugation decreases, HCT decreases. On the other hand, there is another factor called the fertility inhibition factor, FINO, which is a repressor. So if its expression is increased, HGT or conjugation decreases. So we did a real-time PCR on the gene expression of all these genes. We treated the salmonella with essential phytochemicals and extracted the RNA and did a real-time quantitative PCR to study the effect of the phytochemicals on the transcription of different conjugation genes that results in the HCP. So here are the 4 panels, one for transmammalian, carbachol, bacterial, beta-resorcic acid and caprylic acid. To get you up to speed with the orientation here, x-axis are the different genes, y-axis is the expression level of different genes, and black bar is the control, TC, the gray bar, and the control is always because it is not exposed to the basal level is 1. And when we treat it with the TC, you can see that the transcription or the expression of all the TRA genes were downregulated significantly. On the other hand, the repressor genes that we know in presence of transmammaldehyde, its expression got increased. So as I said, because of repressor, if its expression increases, conjugation decreases. We found similar results with the other 3 phytochemicals. Of course, different genes were regulated differently, but consistently we found that the phytochemicals definitely have an inhibitory effect on the genes which express for the pilus and thereby conjugation. We wanted to validate the results, so we did an electron microscopy, TEM. We stained the bacteria, grow the bacteria with phytochemicals, like did negative staining and did TEM image. We wanted to see whether the phytochemicals have any effect on the pilus formation, on the morphology. So these are the control bacteria, salmonella. As you can see, there are a lot of hair-like appendages. These are all the pili, but compared to that, the control, the treated bacteria, so you can see there are substantially lower pilus formation. So we knew that the primary mechanism is through pilus synthesis, and especially by downregulating the genes which contribute to the pilus formation, thereby decreasing HTT. So currently we are doing follow-up studies. We definitely, because we saw the error in the live birds, we are following up, conducting now experiments with the large chicken numbers. And then we are also looking into, because I told you the natural antibiotic resistant flora may have a role. So we are doing a metagenomic based sequence resistome. So resistome means looking at all the resistant bacteria in an environment. So we are looking into that, and also we are going to do the transcriptomics of Salmonella and E. coli treated with the phytochemicals, so that we could detect all the global pathways that are affected by the phytochemicals. So these are the future directions, and so our model is once you reduce HGTR, the poultry gut, poultry environment, their transmission to the humans through the poultry meat and through direct zoonotic effect could be reduced. This is the working model or our hypothesis. And then finally, I will conclude my presentation with a story. Actually, it is not a story, it is a real thing happened. You know that in the 1960s, the Cold War between the United States and USSR. Of course, now we don't have it, and some of you may not know even USSR, because after perestroika, USSR disintegrated into many countries. The major one is Russia. So when the Cold War was happening, both of them were in competition, the two countries, who will be putting a man in the moon for the first time. So a lot of work was going on in NASA, and they were really taking the time to get ready to put a man on the moon. And so President Robert Kennedy visited NASA, which is based in Texas, just to get an update, discuss with the engineers, scientists, to see where they are. During the discussion, after the discussion, he took a walk through the hallway. Then he could see, he met a janitor. Janitor is a man who cleans the place, right? An old guy. So he asked him, "What are you doing here late midnight?" So the janitor, the president asked, the janitor immediately said, "I'm helping NASA to put a man on the moon." It looks like a very simple gesture, statement, The moral is, no matter how big a small job somebody does, in an institution or in a project, everyone contributes to the success of that project. I recognize it being an administrator in my university, and also especially with my huge $10 million project. Although I initiated the project and served as the project director, the collaboration from everyone is critical for the success of the project. So that's the moral of the story. Thank you for your attention. Happy to answer the questions.
It's a wonderful presentation, sir.
Thank you.
And also it's a new idea. When you are talking there, you told you have a different kind of approach.
Yeah.
There it's wonderful to see the kind of approach you are having. Usually we think that antibiotics means protein inhibition, killing the bacteria. Only in that way only we are tuned to think.
Yeah.
But you are talking about how the transfer of the antibiotic resistance is taking place. In your mechanism, I have few doubts there. In the mechanism what you have explained, you have just targeted only on salmonella.
Yeah.
What about the E. coli?
Yeah, actually, the reason we started with salmonella, of course, we are repeating the same experiment in E. coli. The reason we started with salmonella, this was completed just a few months ago, because that's a part of the big project, right? So we were working on it. Because salmonella being the multi-drug resistant bacterium, it has the BRCA gene, so it is the donor. Without a donor, this cannot happen. Bacteria, two normal bacteria also conjugate for sharing genetic information, other information. But we wanted to first see whether we could block it at the donor level. So that's why we used to study the mechanism. Of course, that's a very good question. We'll be following the same with E. coli. And as I said in my concluding slide, the transcriptomics would reveal all the pathways, both in E. coli and salmonella, to see all the mechanisms which are affected by the treatment with the phytonutrients.
Then I have another basic doubt. How common this between the genus this transfer of the genes?
Between the genus, between species, and between serotypes, it happens, as I told you, we had lactic acid barrier bacteria, Lactobacillus acidophilus, Lactobacillus plantarum, Streptococcus lactis, or Lactococcus lactis. These are the three probiotic bacteria which we commonly find. So I never expected that they would receive the antibiotic resistant gene from the Salmonella hydatha. Even in the food environment, 4 degrees centigrade refrigeration, in milk sample, in meat, and then give out to other bacteria.
Yeah, it's a revelation for me.
Yeah.
Thank you very much.
Yeah.
Again, congratulations for the wonderful talk which you have delivered, sir. One small question is, now, as you told, there are 3 methods which are responsible for hydrogen gene transfer. Now you are mainly concentrating on conjugation, and as your mechanism says that it is inhibiting production of pili. What about the other 2? Though they may be minor, but they are also responsible for transfer of resistant genes.
Yeah, of course. Especially the antibiotic resistant, the literature says the antibiotic resistant gene transmission is heavily through conjugation, because of the nature of the genes, and that the cells have to be competitive, and that they have to. It can happen through virus. We know that bacteriophages attack bacteria, they take some genes, transfer it randomly to other bacteria. It could happen. But mostly those are reported to be mostly virulence genes. We have not, there could be, and transcription, transformation is mostly, we transform the bacteria in the lab, right? Transformation usually for antibiotic resistant genes happens when the conditions are very favorable. You have to provide really, when we electroplate in the lab, we had to give the actual very conducive environment, 27 degrees centigrade. We had to add some minerals to increase the. So it could happen, but mostly antibiotic resistant has essentially. And we cannot, the phytochemicals, of course, some phytochemicals have a antiviral effect, but this is not a way to target.
Thank you.
Any other questions? From the students, look forward to questions from the students. Don't have any inhibition. When I was sitting on your side, when I was a student, I was reluctant to ask questions. Well, my questions would be foolish. How the others would think? Never have that thought. Ask any question that comes to your mind.
Sir, good morning, sir. I'm Gaurav Ragan, PG student of here, sir.
Okay.
Thank you for the wonderful presentation, sir.
Thank you.
Big work, sir. My doubt is on the first slide you have put, sir. You said the antimicrobial resistance was developing due to peak diseases, antibiotic is spreading to the disease and the gaining bacteria are gaining antimicrobial resistance. How could we stop that, sir? As for environmental, we have to, because 40 to 60% of the antimicrobial resistance are developing from that. What is the procedure or what is the process we have to do for that?
For the manual or the environment, interventions at the environment. So that's what, ultimately what we are going to do is, we are going to do this in a huge manure pot. So treatment with the manure with the phytochemicals could break that spread from one bacteria, which what we demonstrated in the tubes with fecal matter, we could take it to the next level. So the ultimate application is in a manure, because if you have visited the poultry farms, especially big ones, they have huge piles of manure. So treat that manure or with phytochemicals or even water and with the phytochemicals. So this doesn't happen and it doesn't get stored. I hope I understood your question and answered it right.
Other than phytochemicals, is there any possibility for the
Of course, that's a good question. There are certain methods at very low concentration. There is a study which was published, selenium, at very low concentration has been shown to disrupt HGP in sewage systems. We are looking into probiotics that would, the whole study started whether probiotics would inhibit HGT, then we found out that probiotics ain't the resistant genes. So we are looking into other bacteria, whether are there any bacteria that could destroy.
Okay, thank you.
Any other questions?
Hello, sir. Sir, for the phytochemicals,
Could I have your name, please?
I'm Naina. I'm an intern student.
Okay.
Sir, for the phytochemicals, like you have extracted them from the source, and then have you tried like instead of extracting them from the source and like directly giving the coconut oil to the poultry? Because like what if farmers give directly coconut oil to the poultry and like will it be effective, and what would be the comparison of efficacy of both of the two?
Yeah, that's a very good question actually. For your information, I did not use the original. We have companies which extract this and we get it in pure form. Molecular biology grade 99%. So that's what we use because as a controlled experiment, we have to show the proof of principle using the pure molecule. To answer to your question, if we use coconut oil, we have tried that. What happens is coconut oil has a lot of other components, a lot of other chemicals. So what we have seen, the availability, this is what the hypothesis when I collaborate with the organic chemists, the availability of that particular caprylic acid to target the bacteria is diminished. So we have seen when we give coconut oil to chicken, we don't see a big effect in terms of antibiotic, not antibiotic resistance, in terms of pathogen production. So, in my experience, what we work, and many people use plant extracts actually, forget about coconut oil, extract from the plants. Even that has hundreds of molecules, chemicals. What we have seen, the targeted molecule we want, its efficacy is not equivalent to a pure molecule when we use.
So is it still effective?
Pardon?
Is it still effective?
It's very minimal effect in my experience we get when we use, there are lots of purpose people are used extract from plants for different activity. There is some effect, but when we finally end up test it in an in vivo system in animals, I don't see a huge effect.
Thank you, sir.
Influence.
You can ask questions, anything else outside this topic, I'll be happy to answer. If you don't have any questions on this topic. Okay.
Thank you, sir. Thank you, sir, for taking time and sharing your valuable insights. We're incredibly privileged to benefit from the wisdom shared by you. I now request our beloved Dean, Dr. V. Sayan, to say a few words.
Good morning, everybody, and thank you so much for the wonderful presentation. Because I thought River is keeping its foot on the AMR now. It's one of the buzzwords across the globe. Very timely talk as echoed by Professor Tanis, because a lot of basics have been cleared. Maybe it could have been little over and above for all of you to understand some, but then you see, you should understand, being a youngster, what Dr. Kumar is trying to impress upon all of you is that you need to choose your pathway, because his presentation has so many messages for you. Even if you are not understood, that's what he meant. Even if it is out of context than the talk, you could have interacted in a much better way. I always felt, these are the opportunities the students should never miss. Because such a world-renowned scientist he's been, and down-to-earth scientist, and we should encash that by his presence here, because it's not going to happen very frequently to have him like that, and then we should make an use of that. You see, today, the youngsters sitting here, especially the PG students, the front runners now for the career, as far as the professional career is concerned, you need to choose your pathway very clearly. That's what his message is. See, his concluding slide is spoken two things very important for the future: metagenomics and transcriptomics approach. These are the clues that you people should take, and then you should ask queries related to that. How to shape up your career using metagenomics or transcriptomics, because these are all path-breaking biotechnological tools that are available with us currently. We can explore getting answer for all the hidden things that we have never explored till date. So these are the pathways that he's been projecting. And also, today globally 4 or 5 topics are very burning topics where you can have a very good future. For example, AMR stands first, and also animal welfare, climate change,
food security. These are the aspects that we need to concentrate on. Because if you wanted to see why people are in limelight, for few scientists are not in limelight, it's all about the area you choose with, and then that gives you a name and fame and a sound opportunity for getting quality publications, everything. So these are the type of messages that you need to take from this type of talk. And then I would like my students to be more interactive, because like we don't get to such an opportunity very often. So even that, let it be very, very simple. See, that's how we have developed. It's not going to be overnight that we can produce quality students. It's all about a process. That is, you all been put, and these lecture series are primarily meant for that. You have been assigned a duty today, and then we have taken permission for you guys to come here and sit and listen to the talk. And these are all very basic and fundamental concepts we need to concentrate really hard on. So it's like this is a message for all of you for the future, because you need to choose your future, because it's important. I was surprised, like a lot of new young bunch of meritorious microbiology students, PG students are sitting, and then no questions comes from, which means he has taken it very clearly. We know that we have understood very, very clearly, crystal clear is the explanations were. You could have understood much better, at least from the future point of view. You could have asked something else. I also happy to share that he has come all the way, not for this talk, but then we had an important agenda yesterday. Like we have signed a formal MOU with the University of Connecticut yesterday, and it's a very great way forward, because the support we got from Connecticut for last 2 decades is immense. With that view intention, like we have signed an MOU now, I would like to make a note here for Dr. Kavades sir as well as Dr. Bandaka madam, that see that now we are starting the PG program in a big way, and with the establishment of CTR, the research have to be at its peak now. So it's important that as we have done it for Dr. Binuni, these are the type of scientists that need to be in the advisory board of the students. Like now with the formal MOU already signed, there is a huge opportunity for us, because this team has a very, very good, right from Roshni until Hindu. They are all there having several million projects running up. So it's important that we need to rope in such type of co-supervisorship or something like that, because as we have promised to these students, there should be a foreign mentor in sport as in the advisory board. So these are the opportunity to be encashed, and with the MOU signed, that is also one of the way forward to take the collaboration forward, because we need to have joint publications for the students for the future. In that way, I feel like his presence is highly significant. His support to this institute is immense, and today we stand here, sir, to thank you from the bottom of our heart for coming all the way, although you have a very grueling schedule last 20-25 days in India, but still you made it, and then we are very happy that we are progressing forward with the type of MOU we signed. And also 4 students are slated to visit you in the next couple of months at the University of Connecticut for the externship program. This is the way forward, and then we wanted a lot of supervisorship, co-supervisorship and other type of collaboration before we start on the research front, and some international collaborative projects both institutes should hold. So this augurs very well for our future. With these few words, I once again thank all of you, especially the head of the departments, the core group who is going to work on AMR in River, and especially in the poultry sector, and as well as my PG students, as well as the undergrads who are sitting over here, and taken inspiration from this type of the talk. He is an Indian. He has made many people proud being an Indian, and is a source of inspiration for many students for the younger generation. That is the message that we all should carry from this. And thank you so much, sir, for once again for making it all the way. Your valuable presence. We really look forward for future collaboration. Thank you so much.
Thank you, sir. As we conclude this enlightening session, I take this opportunity to express our heartfelt thanks for sharing his immense knowledge and expertise with us, and to give us an insight about the use of phytochemicals to reduce the horizontal gene transfer. And I appreciate all the attendees for their participation. I hope everyone leaves today with a greater understanding and new perspective on today's topic. Thank you.